Today's lesson is on tissue fluid, but just as a little introduction, I want you to think about what blood is. So, what is blood? Yeah, hopefully you've thought about it and you've realised that blood is a red liquid that is carried in blood vessels around the body. It comprises of a solution called blood plasma, which carries many solutes such as amino acids, glucose, fatty acids. It contains lots of ions and hormones, and the solvent is water. We've also got red blood cells and a number of different white blood cells, and you've looked at a few of those in the reproduction immunity topic, and there are also some blood proteins. So albumin, I think, is one of them, fibrinogen, and we've also got platelets. So the blood is plasma, which is a solution, white blood cells, red blood cells, platelets, and large proteins, large insoluble proteins. Okay, tissue fluid. So what I want us to achieve by the end of this lesson is to be able to describe what the function of tissue fluid is, to describe how it is formed, and also apply the idea of tissue fluid formation and removal or drainage to various health issues. So, tissue fluid bathes all cells. It is the medium with which substances are exchanged. So, actually, if we're talking about the cell receiving essential substances, they're actually diffusing from the tissue fluid. It comes from the blood originally, but it goes via the tissue fluid. The tissue fluid is produced from blood plasma. It has almost the same composition of blood, apart from the fact that it doesn't have large proteins in it, which you do have in the blood. Carbon dioxide dissolves into the tissue fluid from the cells. Well, it will be dissolved in the cytosol and then diffuse into the tissue fluid. And oxygen diffuses from the tissue fluid into the cytosol of the cell, which is the fluid part of the cytoplasm of the cell. So here is a diagram which looks a bit complicated. I'll just get my pen. Go with red. See if it's working. There we go. So, what is going on here? So this is the arterial end of the capillary. So the arteries or arterioles are supplying this branch off into these capillaries, and the blood here is at a high pressure. Okay, so I'm going to put high pressure. And there's a key word that we want to use here. It's called hydrostatic pressure. Hydrostatic pressure is the force exerted by fluids, basically. So we're at high hydrostatic pressure. Why are we at high pressure at the arterial end? Well, the blood has come via the arterioles and arteries from the heart, the left-hand side of the heart, which has, if we talk about the rest of the body, which has thick muscular walls of the left ventricle. When they contract, they cause a huge amount of force and therefore cause the blood to be under high hydrostatic pressure. You've also got to think about what we did last lesson, and the structure of the arteries and arterioles maintain that pressure. So as soon as there's a drop in pressure, because we're at diastole, which means the relaxation of the heart, the elastin in the artery walls causes them to recoil, decreasing the volume of the artery to maintain that pressure. Okay, so the pressure is kept high due to specific structural adaptations of the arteries. Okay, so now it enters the capillary. So net hydrostatic pressure and osmotic pressure. Osmotic pressure is just saying the pressure which is caused by the difference in the water potential. Okay, so we've got a much higher hydrostatic pressure that forces water, ions, glucose, etc. out of the blood into the area surrounding their cells, and it does this between the endothelial cells. However, there is a pressure, osmotic pressure, because the water does want to move back in due to osmosis. But actually, because the hydrostatic pressure is much higher than the sort of water potential gradient, it causes water to move out. As we move further along the capillary, we've lost water. If you lose water, then the water potential inside the capillary is going to go down. So it's going to have a much higher osmotic pressure. Also, if you lose water, you're not going to have as much hydrostatic pressure. So the loss of the water and the fact that inside the blood capillary we've still got big proteins, big blood proteins which lower the water potential, and they're too big to get out between these endothelial cells. So therefore they stay in. So now we've got a very low water potential inside the blood capillary, and we've also got a not as high hydrostatic pressure, slightly higher hydrostatic pressure. So now the water wants to move back in. So water moves out due to hydrostatic pressure, and then water moves back in further down the capillary where it's just about to join up to form the venules. Water's moving back in due to osmosis. So arterial end out due to high pressure. As we move along the capillary, water moves back in at the venous end due to osmosis. That's the key points that you need to remember. So, with a lot of biology, there's prerequisite knowledge that you need to have. So, what knowledge do you need to have to be able to really understand tissue fluid? So I mentioned things like pressure exerted by fluids on the walls of the vessel. You need to know about osmosis. Luckily, we did osmosis in detail at the beginning of the year. What else do you need to know about? So I also referred to, let's turn this off. I also referred to the structure of the arteriole, the fact that it's got elastin in it. That's what we did last lesson, so you'd need to be carrying the knowledge on from last lesson. You need to know the composition of blood. Now, we've not gone over that in much detail, but you did learn it at GCSE. We have to know about why oxygen and glucose would be in the tissue fluid and why they would be diffusing into cells. Well, both of those are actually used up in aerobic respiration, so therefore we've got a concentration gradient where we've got a higher concentration of glucose and oxygen in the tissue fluid, so it will diffuse into the cells. And again, this idea that fluids and gases and liquids can be called fluids will always move from an area of high pressure to slightly lower pressure down a pressure gradient. And we've seen that time and time before through the course. Most recently when we've looked at ventilation, how we can change the pressure in our thorax. Okay, this is quite a detailed description of what is happening. It's kind of what I've just been saying, but we'll put these in or go through them. You might want to stop the video and actually copy this down, maybe draw it as a flow diagram. So, the first one is, they are actually in the right order. Blood enters the arterial end of the capillary and it's at a high pressure. The higher pressure is maintained in the arteries due to the elastic tissue. The higher pressure in the arteries causes a high hydrostatic pressure. Let's cross that out. We don't say plasma is forced out. I need to change that. The water is forced out. Never say the plasma is forced out between the endothelial cells. the blood proteins are too large to fit out. Next one, as plasma leaves the capillaries, not again, as water leaves the capillaries and ions and glucose, the hydrostatic pressure in the capillaries drops. The blood proteins have remained inside the blood, so therefore the water potential of the blood is lower than the water potential of the tissue fluid. The sugar in the tissue fluid diffuses into the cells where it's used for respiration. Amino acids and fatty acids will also move into the cells through facilitated diffusion, where they might be used to synthesize new molecules. So therefore there'll be a concentration gradient where there is a lower amount inside the cell because they're being used up. The water potential of the plasma is lower than the water potential of the tissue fluid. The hydrostatic pressure is higher in the tissue fluid because we've lost water from the blood capillaries. So water starts to move back in at the venous end. And then finally, not all the tissue fluid is actually returned to the circulatory system, straight back to the capillaries. The rest is drained into the lymphatic system, and lymph is actually eventually travels around the lymphatic system in the lymph vessels, and it's returned to the circulatory system via the subclavian veins. Okay, and this is a diagram to help you understand. So this is the arteriole, which branches out into the capillaries, which then, this is where the loss of water and nutrients happens, where the tissue fluid is formed, and then the capillaries at the venous end, you've got water moving back into them to make venules and veins, and then we'll go back to the heart. These green vessels here, these are the lymphatic vessels, so they'll be draining some of the tissue fluid. What actually happens is if there's any sort of bacteria or viruses around these cells trying to get into them, they'll also get drained with the tissue fluid into the lymphatic system. And if you look here, these are little things called little nodes. So a lot of the tissue fluid gets drained into these vessels along with everything else surrounding the cells, such as pathogens, and they'll end up in these little nodes. And in these nodes you've got a high concentration of immune cells. And so if there is inflammation of these nodes, it means that there's probably an infection, or your white blood cells have detected some pathogens. So quite often you can tell if you're actually suffering from a viral infection if you've got, you can feel just underneath your jaw and feel these might be swollen. Okay, and it's a good sign to say that you're actually fighting an infection. So this is the lymphatic system. You don't need to know any other detail about it. Okay. So have a go at the questions and let us know how you get on. Thank you. Bye.